All right, my name is Trinity Martin, and I'll be presenting for the drug discovery class. Our lab was on Drosophila melanogaster as a model organism for identifying potential chemotherapeutics. First off, I'll be going over cancer as well as the treatments and cancer statistics. So about 50% of Americans will get cancer within their lifetime. And so we want to look at kind of what are the causes. The causes are not only environmental factors, but genetic factors. And as you can see in the chart, it's a lot of environmental factors. So it's a lot of how you live your life and what you decide to what you decide to do with your body and how you can help that way. But sometimes it is genetic, so it can't always be stopped. If we move on to why we study cancer because in 2019 alone, 606,000 people died from cancer, and then 1.7 million people were diagnosed with cancer. The side effects are not only super dangerous, but chemotherapy, as right now, isn't super effective. Survival rates for specific cancers are still pretty low. If you want to look at the chart over here, you can see on the x-axis is the year, and then on the y-axis is deaths, and then each of the lines is a different type of cancer. So for like lung cancer, you can see with the popularity of smoking, the cancer went up, and then as the popularity of smoking came down, the line went down. But if you also look at something like pancreatic or liver cancer, those lines are virtually straight, which is really sad because we've been doing research for almost 100 years. You would think we would have something to help those people with liver or pancreatic cancer, but so far, like, we just haven't gotten there yet, which points to more cancer research. More cancer research is something we can do, as well as combinatorial therapy. So that's radiation with chemotherapy. The benefits there are it's overall less toxic because you can use a lower dose of chemotherapy if you're using radiation with it. And it's sort of like fighting cancer with two weapons instead of just one. Next, I'm going to move on to our experiment as a lab, as well as methods and design. So as our model organism, we used the Drosophila melanogaster, which is our fruit fly. They were a really great organism to use because their rapidly dividing cells are very similar to the way a cancer cell will work. They're also super responsive to treatment. They have a pretty short life cycle, and they're pretty inexpensive to use. So overall, they were a great organism to use. So we had an overall hypothesis throughout our class, and that was that 50% of the Drosophila would die from radiation because the majority of our Drosophila were treated with radiation, so about 50% would die from that. And then colchicine was our positive control. So that's a known chemotherapeutic, so you would assume that it's going to work. And with radiation, you expected a 0% survival rate for that test. Onto our methods. So for our methods, we placed adult fruit flies in population cages with grape juice agar plates in the hopes that they would lay embryos on those agar plates. Then we would collect the embryos from the grape juice plates and transfer the embryos to fresh bottles. Then we would collect them by a specific size. We wanted third instar larvae just for the experiment, so that way all of our larvae were similar in age. Next, we would irradiate this third instar larvae, and then we would transfer that larvae into our food and drug mixture. So it not only had food, but it had our whatever compound was being tested as a new chemotherapeutic. After that, they were incubated for 10 days at 25 degrees Celsius, and we were able to quantify survival after that by just seeing which larvae came out of their pupa and which didn't. What we were trying to do is identify hits. Hits are anything in these light blue colors on this graph, anything that's a radiation protector, so less than 50% died or way more died. Obviously, that's going to be a possible chemotherapeutic. So that's how we were able to identify what we wanted. Unfortunately, my personal project didn't work, but there were a couple projects that did have hits, so we'll be going over those. Next, we'll be going over interesting results from other projects. So this project is really, really interesting. Obviously the word is a little bit difficult today. It's dihydroxyartemisinin and wormwood. So the DHA is actually a compound that comes from wormwood. So the test was to see if that's the factor that was fighting the cancer cells, because wormwood has showed promise in the past. But actually, if you can look in the graph, the wormwood actually killed more than the DHA did. So just kind of interesting. It just shows us that we'll need to do more research with the wormwood to find out which compound was actually effective. So obviously we just want to do more research with this one. And then this is the next one. It's aeroplecinin. It's a compound of the sea sponge. This group actually had 3 hits, which is really, really cool. You can see them, they're the lowest ones on the bar graph in the bluish color. This group had a couple interesting points from their lab. They had 8 dilutions, which was the first interesting part. Most of our labs had 4 dilutions, so they did a lot of work to be able to get a lot of this compound to the Drosophila. And then another interesting part was the lower doses actually worked better than the higher doses, which is kind of not what you would expect from the majority of medications. And so for this one, just because they did have hits, we would just once again say more research needs to be done. On to my conclusions. Overall, we would just want to identify the hits and the experiments, which will move forward in the drug discovery development timeline. And then you can see the chart on the left, it has kind of the timeline itself. You kind of start with a lot of compounds, and then it kind of weans its way down to one because it is really, really difficult to get it approved by anything approved by the FDA. And it does take a really long time. So I think that's just shows another reason why it's so great for us to be doing this research, because it's going to be difficult to find something that works. So the fact that we have so many people doing so many different things in this lab to help possibly find a new chemotherapeutic is awesome. Next, I would like to thank Dr. Chintin Su as our lab sponsor, Dr. Pamela Harvey as our professor, and then my teaching assistants, specifically Murphy, Tucker, and Kathy, were amazing, but all the teaching assistants were obviously great in this lab. Our funding came from Lee Nice Wonder, as well as the Department of MCDB and the University of Colorado Boulder. Overall, this lab was awesome, and I just want to thank all these people personally. Were there any questions for me about my lab?
How does your drug of choice that you tested work? Like, what exactly is it supposed to be doing to?
So basically, our Drosophila were like treated as little tumors because their rapidly growing cells were really similar to the way a rapidly growing tumor is going to grow. And so the hope was that we were going to irradiate them. That would kill 50% of the growing cells. And then we were also going to feed them our new compounds. And then we would see from there if it was an effective chemotherapeutic because they were ingesting it. Does that answer your question?
I was kind of thinking more along the lines of, sorry, I was thinking more along the lines of like what exactly it kind of does going into the, like if it just kills to kill the Drosophila or if it is perhaps like inhibiting something.
That's kind of the hope of this lab, and that's how you can kind of take it forward with more experiments as well. The hope is that you don't get a compound that's too toxic, but you find a compound that still stops the growth of new cells. So it's that this is just a very like beginning step in the process of finding a chemotherapeutic. So at this point, we're just looking for something that is able to stop the growth of cells, and then we would later on worry about the toxicity versus actually stopping cell growth.
Thank you.
Rachel, do you have a question?
Yes, you said in the presentation that you irradiated, I think was the term, the Drosophila. What does that mean? Like, what
That's
does
just
that process
radiation.
look like? Oh, okay, cool.
Yeah. Sorry, there's multiple terms. So I know it can get confusing. It definitely was in the lab at times, but that's basically just radiation for the Drosophila.
Okay, thank you.
Sorry, another question. Is the irradiation just to get the Drosophila to start to, I guess, act like a cancer cell, like having that DNA damage and, I guess, replicating cells much faster, or is it just to kind of get it halfway dead to see what's the further effect of the drug?
So basically, instead of just testing a chemotherapeutic like on its own, like just to see how it like reacts with animals, we want to use it with radiation so that way you don't have to go through that extra step of also testing it with the radiation, because if you're going to be using it with radiation anyway, why not begin by using it with radiation? And I think Pam can talk a little bit more on this too.
Yeah, Minerva, you're on the right track that when you're treating cancer, it's often the case that you treat with radiation and a chemotherapy, with the idea that it's really like two different weapons. You're going to damage the DNA of the cell, and then you're going to come in with probably a different strategy to get a good combinatorial therapy. So yeah, we try to replicate that in the lab so that the larvae that we test are treated very similar to how real cancer cells would be treated in a patient.